In genetics, it is important to find genes. If you find a gene, there's a lot of subsequent analyses that one can do. Whereas if one simply knows that a gene exists, somewhere there's a gene that causes this disease, for example, but we don't know where it is, now you're much more limited. The previous video went through ways of, say, genetic mapping, both in humans, but also, say, in fruit flies, where you could find a gene. Well, why would you care if you can find it in a fruit fly? Well, for example, if you can find it in a fruit fly, you could then, send this off to the lab where we want the sequence of this region. And so now we know that this gene, which causes this mutation, it has this sequence. And a probe could be then developed, which would then bind to that sequence. And then you could then use that probe not on fruit fly genes, because now you already know that fruit flies have a copy of that gene. You could use it on human chromosomes, because when we look at the 22 pairs of autosomes and then the sex chromosomes, clearly they have different sizes, they have different banding patterns, etc. And if then you used a probe, which could be radioactive, it could change color when one added the correct reagent, one might see, hey, the probe that binds to this fruit fly gene, it actually then binds to one human chromosome here, the short arm of chromosome 10. It turns out humans have a copy of this gene on their chromosome 10, etc. And so you could then probe a different gene and it might bind elsewhere on chromosome 3. You might find that a probe would bind multiple places if there are duplicates of this gene or homologous genes of a gene family. And so now here is one way in which you can say, I know now not only that humans have the gene, but I know where it is. It's on, there's one on chromosome 12. There's a second one on the X chromosome, the 23rd pair, etc. So that is a way of finding genes. There are other ways, and what you're seeing now is not genetic mapping, where you're using the frequency of recombination to then calculate where a gene is. You're looking at physical mapping that you saw, the probe bound here. In the same way, let's say that in a certain family, there is a trait which we want to study. And so the question is, where is the gene that causes this trait in this family? Well, sometimes you notice that in that family, one of the chromosomes has an abnormality. So chromosomes can have deleted regions, duplicated regions, there could be an inverted region, there could be something wrong with this chromosome. And so while it isn't now certain, it is certainly possible that in this family there was a mistake which affected the chromosome, duplicated a region, deleted a region, etc. And that then is the cause of this problem. So once again, here would be a way in which we are trying to find out where a gene might be. And so therefore, the physical deformity of a chromosome that we can observe might be the cause of this trait in the family. So it was observations of these physical chromosomes that led us to believe that. Okay. I'll get back to this in a second. Another technique of physical mapping is known as deletion mapping. And just as I had said, some chromosomes have deleted regions. Well, if a mutation is recessive, then it means that in order for a fly to then have express a mutation, if it's a recessive mutation, then it needs 2 copies. But if you have a copy of a chromosome which has the mutation, and the other chromosome, which normally would have a a copy of the region which would have a wild type allele, if this would normally then mean the fly would have a normal phenotype because it has a good copy of the gene. If there is a deletion there, if the fly is missing a section, so somewhere there is a blue gene causing a trait which we just discovered in fruit flies. We would like to know where that gene is. Where is the physical spot that it is located on? Well, interestingly, you can go to fruit fly stock centers and order fruit flies. There's all of these different kinds, including deletion stocks, which are missing pieces of a chromosome. And so you could say, all right, so notice here that there are, on the normal chromosome, there's 3 bands, and we order fruit flies which have a deletion in this region, so that now there's only 2. We're missing a region here. And then we could create a heterozygote which has 1 chromosome for this new mutation that we're studying, and the other chromosome has a deletion. And then the question could then be, do we see the mutant phenotype in our heterozygous fly that has the 2 different kinds of chromosomes? If the answer is no, we do not see the mutation, if the mutation is, say, brown eyes. If we don't see that mutation here, it means that this fly has a normal gene, all right, which can compensate for the mutation. Now, if there's a deletion here, that means the normal gene can't be here, because otherwise, because this area is missing, if the blue gene was actually located here, we don't know whether it is or not at this point. If the blue gene was located here, then now that we're missing a section, the fly would only have 1 copy of that gene, and therefore would have the brown eyes. But now we say, oh, okay, so we ruled that out. The gene isn't at that spot of the chromosome. Great, now let's order a second deletion stock with a deletion at a different point in the fly chromosome. And now let's try this again. And so we get another fly stock, and there is a deletion at another place. This time we're missing this section here. At this point, we don't know whether the blue gene is located there or not. We look at the fly's eyes. Are they brown? They are not. So if we're missing this section, okay, if the blue gene was in this section, then now we would only have 1 copy of the blue gene, the mutant form. The wild type form would now be missing. And then the fly would have brown eyes because there would not be a normal allele to compensate for the mutant allele. But then we would say, nope, it isn't there. And then so we would do it again, and we would do it again. And we would run through a number of these deletion stocks, waiting until when we found a heterozygous fly which was expressing the recessive mutation. Normal heterozygotes would not express the recessive mutation unless they were missing the part of the chromosome which had the normal wild type allele. And so it is in this final stock that we get a deletion where we're missing this section of the chromosome. So when we create a heterozygous fly, they have 1 region with the blue mutant allele, but they're now missing the area that has the wild type allele that would compensate for it. And so there is only then 1 copy of the gene. It happens to have the mutation for brown eyes. And so now our fly is showing a mutation for brown eyes, even though it's recessive. This is a way of mapping known as deletion mapping. We've used a series of deletion stocks in fruit flies to isolate where that mutation is. There are others, and I'd like to just mention a couple of other ways of physical mapping. One is, it is possible, actually, let me just show you another video first. It is possible to take cells and fuse them together. Now, the body does this, but this can also then happen in technology. So if you wanted to make monoclonal antibodies, one of the ways that you could do it is you could take a lymphocyte, which makes antibodies, and fuse it with a cancer cell. All right, and then you would have then cells which not only did cancerous growth, but would then make a specific antibody that you want. And so it is possible to fuse cells. This happens in the body, so some of our body cells are actually have multiple nuclei, but then you can also then do it in laboratory settings as well. Now, you could actually fuse human cells with mouse cells. And let me kind of go through why you might do that. So you take a human cell, which has all the human genes, you smush it together with a mouse cell. Now these cells are unstable for a bit because they have so many chromosomes, they don't really match. And as it divides, many of the chromosomes will be lost. But at some point you can get a cell line which stabilizes, which now has a mix of, say, mouse and human chromosomes. Let's say you want to find a human gene. What chromosome is this human gene located on? Well, let's say we take these three cell lines, which are now part mouse and part human. And here are the human chromosomes. A number have been lost. All right, and so these are the only human chromosomes which are in this cell line. These are the only human chromosomes in this cell line. These are the only human chromosomes in this cell line. And then we ask, if we're looking for a gene, let's say there's a way that we can detect its presence when it's present in a cell. Maybe it encodes an enzyme, and we can ask, which of these cell lines has the enzymatic activity for the enzyme that we're looking for. Let's say we find enzymatic activity here and here, but not here. So we don't know what human chromosome the gene is on, but we know that this cell line has the right chromosome. This cell line has the right chromosome, and this one doesn't. And then we could simply then ask, what is the chromosome that is shared by both this cell line and this cell line that is not present here? And the answer is obviously the big chromosome is present in both of these cell lines. This is present in the second one, but not, I'm sorry, the third one here, but not the first. These two are present in the first, but not the third. This big chromosome is present in all of the cell lines which display activity. This is the human chromosome that the gene for our enzyme is located on. So we have just mapped the gene. And so if we wanted to then apply that here, this goes, let's say both of these cell lines then have the activity for the gene that we're looking for. And we ask, what chromosomes do they both share? Not this one. This one's present in this cell line, but not this one. Not this one. This one's present in this cell line, but not this one. It's this larger one. All right, now we know what that chromosome looks like. We see, oh, that's our chromosome 1. So chromosome 1 has that size, that banding pattern, etc. And the same thing here. It is this chromosome here, which is the one that's shared in all of the cell lines which have the gene's activity. So that was chromosome 3 in that scenario. There's something similar we can kind of do with fruit flies, finding the chromosome a gene is on, but I'd like to just kind of go through two last items here before I end with that. One could do what's called a chromosome walk in order to find the sequences in the correct order. So let's say you cut a huge chromosome into all these little pieces, and you ask, all right, here's one piece, and you know that it ends in this sequence. I'm just going to simplify with AT, but obviously you'd need a longer sequence than that. Then out of all of these random sequences, you would say, oh, wait, here is one that starts here. So there's an overlap. So, sequence number 1 ends with the sequence, but sequence number 2 begins with the sequence. But now, if you were to put them together, you know that the correct sequence would be T, 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 A, T, and then A, A, because this section matches this section. You know that this portion comes next. You don't want to sequence the whole thing. I mean, it might just be too big, but you could sequence parts of it and then align them. And then you would say, all right, well, what, then you could ask, all right, so which of these remaining fragments would then bind to this AA region? So, so far we've sequenced up to this point. What comes next? Well, obviously this has a matching section. So all right, so after the AA, there would be 3 Gs, right? Because, this lines up with that, and then this would be the next section. And then we could go on into the next section, and then the next section, and then out of these random sequences, we then determine, I now know what the sequence of the whole DNA segment is. All right. Now, once we know a sequence, we could look for genes. All right. So maybe, from other studies, we know that it encodes this type of protein, so that we could search for a specific, a consensus sequence. Or if we're just looking for genes, we know that if you're going to make a protein, that during translation you need a start codon AUG, which for right here, because now we're in the DNA part, would be read as ATG. And so here you see an ATG, and you could ask the question, does a gene start there? And the answer is, well, probably not, because we could then look at where the first stop codon would be, because we don't know. It could be an ATG, which is the beginning of a gene, or it could just be a random sequence that is not a gene. And so if this were the beginning of the gene, here would be the end of the gene. Yeah, that's probably too short. So even though that could be a start codon, it's not. And so probably that is not a gene. And then we could do that again. So if here's the AUG, then there would be the stop sequence. And it turns out, once again, that's probably too short. Whereas in this third example, at one specific ATG or AUG, then there's a lengthy section before you get to the first stop codon. So this could then be a gene, all right? And then we could ask, all right, well, if this is a gene, is this similar to anything that we see in another species? In which case, then this is probably a human homologue of a member of a gene family, which, say, all vertebrates share or whatever. Once we ask, is this a real gene, we could ask what amino acid sequence would be coded for by such a nucleotide sequence, and then look to see if humans actually make a gene with that sequence. So that is a way of, say, finding a gene. Another way, and I'm just going to end with this because I have a playlist on this topic. And it's kind of involved, and the playlist does go through step by step all of the things needed to know. But if I were to try to describe it here, this overview wouldn't be that much shorter than the playlist would be. And so therefore, I'm just going to kind of mention a couple of key points here, and then refer anyone of interest or my students to the playlist to get through it in great detail. So one of the useful mutant stocks that one can get is a stock which has 4 dominant mutations. So the wings are curly, they have a little curl, they're spread out, they're dikete. The flies have plum eyes, and they have stubble bristles. So we call it curly plum dikete stubble, and those are the abbreviations, all right? And if you get these flies, this stock of flies could help you identify where a new mutation is, because fruit flies have 4 chromosomes, all right? They have the big first chromosome, that's a sex chromosome. They have a little fourth chromosome. There aren't that many genes there. And then they have two good-sized chromosomes, 2 and 3, all right? And so which of these 4 chromosomes has the new fruit fly gene? Let's say you discover a new mutation that makes the body color green or red or blue or something. Where is that gene? Once again, that's potentially important because maybe humans have a homologue of that gene. If you can find it in flies, then that would be the first step in potentially finding it in humans, and there could be real value in that. So if you find a new mutation, where is it? Well, there's a lot to this cross, as I explain, as it's set up. Like, for example, these are homozygous lethals, these mutations. There are inversions of the chromosome which present crossing over, so these flies will always keep the same ones in the same order. But then after you cross your mutant fly to the curly plum dicheat stubble. So imagine you've got a pink fly and you want to know where's the gene that causes this pink fly. So you made it a female with a mutation to a fly which is curly plum dicheat and stubble, all right? And then you look at the offspring. Now, once again, I go through this in much greater detail here, but you have different kinds of offspring. And unless the gene is sex-linked, and I'll get to that in a second, if you're going to mate flies, if you have, say, curly winged females, which also are dicheat. So they have 2 mutations, curly because there's a bend and dicheat because they're spread out. You can only mate curly dicheat females to a curly dicheat male for this to work in the next generation. If you have curly wings and stubble bristles, you can only mate curly stubble females to curly stubble males. You could only mate dicheat plum males to, plum dicheat males to plum dicheat females. You could only mate plum stubble females to plum stubble males. So in the F1, if you want to take it to the F2 generation, that's the only thing you have to remember. You have to mate the same kind of female to the same kind of male. Why would you want to do that? Well, first off, it would be easy enough to see if this was sex-linked, because if the female has the mutation in a sex-linked trait, if she's homozygous, then all of the males would say have that green body. And so when you do this the first time, if all the females, if all the males of the next generation have a green body, this is the F1 generation, you say, oh, that was easy. That is on the X chromosome. But how about if none of the flies of the F1 generation have the mutation? Well, now here I'm going to mate a curly, a curly dicheat male to a curly dicheat female. And then I'm going to ask, do I see the mutation in the next generation? And the reason I can now map this to a chromosome is I know starting off that curly is on the 2nd chromosome, and that dicheat, having the wings spread out, that's on the 3rd chromosome. Let's say that I'm trying to track a new mutation which causes a red body in a fly. What chromosome is it on? Well, in this cross, if I notice that in the F2 generation, some of the flies with dicheat wings, not all of them, but some of them, have a red body, I say, I know my answer, because dicheat is on chromosome 3. I know that. That's why I got these flies, because they have a dominant lethal mutation on chromosome 3 that I can study. The only way that you can have this recessive mutation is if you have 2 genes for it. This red mutation, it can't be on chromosome 3, because one of the chromosome 3s has dicheat on it, all right? So you can't have 2 chromosome 3s, one having dicheat, and then 2 more having the recessive mutation. That would give you too many. So now we say the only way that you could have 2 chromosomes with a mutation for the red body mutation is if it's on chromosome 2, because these two cannot coexist with dikeet, which is on chromosome 3. Once again, I'm rushing this, but this is covered in greater detail in the individual videos. Now, I didn't have to use the curly dikeet flies. I could have used another combination, like curly stubble flies, for example. So in that case, I was able to say, oh, I now know that my gene is on chromosome 3. I'm sorry, it's on chromosome 2. It can't be on chromosome 3 because it can't coexist with dikeet. So I located this mutant gene. I know what chromosome it is on, all right? And so we could then do that here. Notice that Plum is on the 2nd chromosome with Curly. Notice that some of this other mutation, the blue body color, can coexist with Plum, which is a chromosome 2 mutation, but it can't coexist with dikeet, which is a chromosome 3 mutation. So now I would say, oh, this mutation for the blue body color, it can't be on chromosome 2, all right, but since it can't coexist with dikeet, it must be on chromosome 3. I would say, aha, I've identified this mutation's chromosome. It must be on chromosome 3, all right? Now, if I were to find, my body color mutant, say here, this purple one, sometimes it is with the chromosome 2 mutant. So sometimes it is with curly wings, sometimes it is with dikeet. So here, some of the mutants have a chromosome 2 that has curly on it. Some of the mutants have a chromosome 3 which have dikeet on it. And so now I know it must be on this little chromosome 4. Okay, so this playlist goes through this mapping in greater detail. But as a result, you could say, I now know which chromosome my new Drosophila mutation is on. Using that, then, I am now going to do the genetic mapping using crossover frequencies that I described in the previous video to find out where on the chromosome. All right, so here I've mapped it to a chromosome, and then using the frequency of recombination, I can now map it to which part of the chromosome. And once I have that, I can say, to the lab, could you please, give me the sequence of this part of the Drosophila chromosome? I think this is the sequence of the gene that I am looking for. So there are a variety of ways of trying to find genes, and this was an introduction into some of them. Once again, this final portion was a bit rushed because it's involved. So I have a playlist that goes through at a much slower pace, mapping using the curly plum dikeet stubble flies.